Good morning students, hope you are doing well. Today we are going to study about vector systems. Before we go to the details of vectors, we have to revise some lessons which we have already studied, basic steps in gene cloning. I hope you remember this topic. There are 5 basic steps in gene cloning. The first step is construction of recombinant DNA. The second step is transport into the host cell. Then the third is multiplication of recombinant DNA molecule. And the fourth step is division of host cell, and finally clone formation. So in order to construct a recombinant DNA molecule, first of all you have to select your gene of interest, then you will have to find out a suitable vector system. Once when you insert gene of interest into the vector system, it will become a recombinant DNA molecule. Because these two things are from two different sources only. Now this is a recombined DNA molecule, so it is a recombinant DNA molecule. Then this recombinant DNA molecule will be transferred to the host system. When we transport this into the host system, we will call the process as transformation. Here a bacterial cell is shown, its chromosome is shown here, and this is the plasmid, like this is your vector. The plasmid is constructed as a vector. Then the third step is multiplication of recombinant DNA molecule. Within the bacterial host system, this vector will get replicated and you will get numerous copies of your vector molecule. Then after some rounds of replication, obviously the host chromosome also replicates once, but your vector can replicate many times. So the cell will go for division. Now you are getting 2 progeny cells. After so many rounds of replications, you can transfer a part of your culture into a petri dish. Like you are inoculating an aliquot of this culture on petri dish with nutrient medium, and you will see, obviously you will observe colonies. And for a microbiologist this is a colony, but as a genetic engineer you will call this as clone. You know, clones are, the meaning of the word clone is true copy. So the members of a particular colony, they are having identical genetic makeup because they are all arised from a single cell only. So now your recombinant DNA molecule is part of the colony. Every member of this particular colony carries your recombinant DNA molecule. Some number of copies of your recombinant DNA molecule is present in every cell. So the basic idea of gene cloning is to propagate a gene of interest to a great number, and afterwards it will get expressed within the host system and you will get the protein product from that particular gene of interest. So when we go for constructing a recombinant DNA molecule, our intention is to find out maximum number of recombinant DNA molecules, but that's not the case always. You know, in your reaction tube you have vector DNA molecules and you have to cut open your vector for inserting your gene of interest. This gene of interest should be incorporated into the vector system. For that you have to cut open your vector DNA molecule. And you know how to cut open this molecule. To open your DNA molecule, you have to use some restriction enzymes. So here I have marked restriction enzymes. So this is vector plus restriction enzymes. So when you treat your vector with restriction enzyme, it will be opened at a particular position, and now it will have some sticky cuttings. Like in a particular molecule only one cutting, it's get opened. So now you will introduce your gene of interest. You want to see that your vector is converted as a recombinant DNA molecule, but there are 2 options. Like sometimes your gene of interest will become the part of the vector molecule, thus you will get a recombinant DNA molecule. But there is equal chance that your vector can self-ligate or re-ligate. Like you opened these 2 cut ends, they are sticky ended. These sticky ends are compatible each other. So any time they can re-ligate. If it gets re-ligated in that way, then you will say it as a self-ligated DNA molecule. Or there is another term, unaltered vector. Self-ligated or unaltered vector means the vector remains as such. It was just got opened, but as the cut ends are compatible each other, they went for self-ligation. These 2 cut ends, they join each other without inserting your gene of interest. But what you want to see? You want to see that your gene of interest has become the part of the vector, because only recombinant DNA molecule will have some use for you. Now, So in the we cannot help it. In the reaction mixture, we have two different kinds of molecules. You have recombinant DNA molecule, that is of your interest, and then you have self-ligated or unaltered molecule, but you cannot see them. So what you will do? Now you will go for, like, you cannot separate them at this level. So what you will do? You will go for the second step, transport into the host system. So this is your bacterial cell culture. Number of bacterial cells are there. Now the bacterial system has also different options. Like some cells, they will receive your recombinant DNA molecule. The cells who receive recombinant DNA molecules are known as recombinant cells. Some of them will receive these self-ligated DNA molecules. If they receive self-ligated DNA molecules, then they are non-recombinant cells. And some of them, they will receive nothing, then we will call them as non-transformed cells. Or we can say there are mainly two different categories of cells: non-transformed cells and transformed cells. Transformed cells are of two types. They can be recombinant or they can be non-recombinant. So the main categories are two, like non-transformed cells and transformed cells. But transformed cells are again two. First category is recombinant cell and the second is non-recombinant cell. Okay. So there are three different types of cells in the cell culture. And you are interested only on recombinant cells. You are not interested on non-transformed cells. You are not interested on non-recombinant cells because these two are not carrying your gene of interest. Recombinant cells are the one with a plasmid which is constructed as recombinant DNA molecule. Non-recombinant cells, they received something. That's why we call them as transformed cells. But they have received, that is of no use for you. And then the next is non-transformed cell. Non-transformed cell was not ready to receive plasmid from the mixture. So now you have three different types of cells and you have to screen this cell culture for finding out your recombinant cells. Because you have to exclusively subculture recombinant cells to propagate your gene of interest. So now we are going to study the vector systems one by one. The first vector system is pBR322. P stands for plasmid, BR stands for Bolivar and Rodriguez. These are the two scientists who developed this particular vector system. And 322 is the number which distinguishes this particular vector from all other vector systems developed in the same lab. We can see now this is an artificial cloning vector. We generally use artificial vectors and artificial molecules as cloning vectors. What's the reason? We have already discussed this in our previous classes. Like natural plasmids having lot of negative aspects. Because of that, we will go for constructing artificial molecules as cloning vectors. So we can see this is the most popular cloning vector. The simplest and the most popular cloning vector is pBR322. We can just see the features of this particular vector system. It has an origin of replication. We learned that origin of replication is the first and the foremost requirement for a vector system. And the second feature is it has selectable marker genes, ampicillin resistant gene and tetracycline resistant gene. These are antibiotic resistant genes. These antibiotic resistant genes will help us in the process of selection. You know now what is selection? We have three different types of cells in our cell culture. We have to find out who is who, and we are interested only on recombinant cells. So for the selection of recombinant cells, these selectable marker genes will help us. And now the size of this molecule is only 4363 base pair. Only 4.3 kb inside such a tiny molecule. And see the features: size is 4363 base pair, 4.3 kb in size. Copy number is 15 to 20 per cell. In a particular bacterial cell, you will see maximum 15 to 20 copies. And then two selectable marker genes are there: ampicillin resistant gene and tetracycline resistant gene. Okay. So its features are quite simple to understand. Now, ampicillin resistant gene is derived from PSF2124. Tetracycline resistant gene is derived from PSE101. Origin of replication is derived from PMB1. I'm sorry, it's not clear here. PMB1. PMB1 derived. So what are these things like PSF2124, PSE101, PMB1? These are natural plasmids only. So an artificial cloning vector is constructed by taking out some particular segments of DNA from natural plasmids. So PSF2124 is a natural plasmid. From that plasmid you have got your ampicillin resistant gene that you kept in this particular molecule, and tetracycline resistant gene is also taken from a natural plasmid, that is PSE101, and origin of replication is taken from PMB1. So now we have engineered a molecule, or we have artificially constructed a DNA molecule, a viable DNA molecule that can carry your gene of interest to a host system, and the host will help us to propagate our DNA molecule, like your gene of interest inside the host system. Okay, so now we can see like where we will insert our gene of interest. Now you have ampicillin resistant gene and tetracycline resistant gene. A number of restriction sites are present on these two selectable marker genes. Actually, there are approximately 21 restriction sites throughout this particular molecule. But we prefer to use the restriction sites which are present in selectable marker genes, because when you insert your gene of interest into a selectable marker gene, the property of the cell which receives that particular molecule will change. Phenotypic property will be changed. That's why we want to insert our gene of interest to any one of the selectable marker genes. And conventionally, people use BAM-H1 site present in tetracycline resistant gene. You have equal right to use all of these different restriction sites. But generally, people use BAM-H1 site of tetracycline resistant gene. So now I just show, suppose this is got opened, like with the help of BAM-H1 enzyme, you opened this particular vector DNA molecule. So there is a cleavage in the tetracycline resistant gene, and you are inserting your gene of interest into tetracycline resistant gene. Okay, gene of interest is inserted here. So now what is happening? Your tetracycline resistant gene is no longer a tetracycline resistant gene. In the reading frame of tetracycline resistant gene, now there is your gene of interest. So it will not code for its own protein. Tetracycline resistant gene cannot code for its own protein. When the cell uptake this particular recombinant DNA molecule, it will go for coding the protein from gene of interest, not from your tetracycline resistant gene. Okay. So now we can see, like in the previous slide I had shown you, there are two different types of molecule. When you go for constructing a recombinant DNA molecule, either you will get a recombinant DNA molecule, not either or, like we can say both the type of molecules will be there. Like you will be having recombinant DNA molecule in the reaction mixture, at the same time you will have self-ligated or unaltered molecules also. Our challenge is to find out the cells who are carrying the recombinant DNA molecule. Okay, so now what is the selection principle? Selection principle is insertional inactivation of tetracycline resistant gene, and selection strategy is replica plating. So we can try to understand this. The first thing is the principle, insertional inactivation of tetracycline resistant gene. Already we understood that the biggest challenge in this process, in gene cloning process, is to find out the cells who are carrying your gene of interest. Obviously, the gene of interest is with your recombinant DNA molecule only. So the cells who are carrying recombinant DNA molecule, they must get separated from other cells. So insertion inactivation of tetracycline resistant gene will help us in this process. But how will we know like tetracycline resistant gene is got inactivated or not? For that we have a selection strategy, that is replica plating. Okay. So we can say what is this replica plating process. So in like in the other chart, you have seen that when you go for transferring your reaction mixture, like after the construction of recombinant DNA molecule, you will just transfer a part of that reaction mixture to a conical flask where bacterial cells are there. And bacterial cells have three different options. Some of them, they will receive your recombinant DNA molecule, some of them will receive your self-ligated or unaltered DNA molecule, and some of them will receive your they will receive nothing. They are non-transformed cells. Okay, so from that conical flask, you will go for transferring an anicot to a petri dish. Some part of that culture that will be inoculated on a petri dish containing nutrient medium. So here, the process of selection that will take place in 2 days. On first day, what you are doing, you will prepare a nutrient agar medium along with the antibiotic ampicillin. Okay, and now you are transferring your culture onto this petri dish. Okay, some 100 microliter of the culture that will be spread on this plate with the help of an L rod, and colonies will be observed next day. Like after overnight incubation, you will see colonies. Okay, so now can you please tell me like how many different types of cells can grow on this plate? We have totally in your culture flask, you have 3 different types of cells. And who all can grow on this plate? This plate is it put forward a particular kind of selection pressure. What is that selection pressure? In the medium you have put ampicillin antibiotic. So the cells who are surviving on this particular plate, they are definitely antibiotic resistant. Which antibiotic? Ampicillin resistant cells only can grow on this plate. So how many different type of cells can grow on this plate? Only those cells who harbor a plasmid with antibiotic ampicillin resistant gene. Ampicillin resistant gene should be present in the plasmid. Then only it can grow. The cells who harbor the plasmid with ampicillin resistant gene, they can only grow on this plate. Okay. So now can you tell me like which all can grow here? Definitely transformed cells can grow on this plate. Because transformed cells have got a plasmid, and in the plasmid there is ampicillin resistant gene. So definitely transformed cells can grow on this plate. But one more thing, your non-transformed cell, they haven't received any plasmid. They are non-transformed. They haven't got any foreign material. Matlab, they don't have antibiotic resistant property. Antibiotic resistant property is conferred by the plasmid only. So now the transformed cells can grow here, but transformed cells are of 2 types. One category is recombinant cell and the other category is non-recombinant cell. And you are interested only on recombinant cell. Why? Because recombinant cells are the one which carry your recombinant DNA molecule. Recombinant DNA molecule means your gene of interest is the part of the molecule. Okay, so recombinants and non-recombinants both can grow on this plate as they are transformed with plasmids, and that plasmid carries antibiotic ampicillin resistant gene. Because of that, like ampicillin resistant gene, what it will do? It will go for coding an enzyme known as beta-lactamase, and your antibiotic ampicillin has beta-lactam ring that will be inactivated with beta-lactamase enzyme. That's why the cell is capable to grow in the medium containing ampicillin. But for that, this particular resistant gene is required. That will only code for the enzyme. Then only, like that enzyme production must be there. Then only it will grow on the medium containing ampicillin. Okay, so this part is clear. Like here we have both recombinant cells and non-recombinant cells. I'm just marking these two as recombinant cells. This I'll say as non-recombinant cells. These two as non-recombinant cells and these two as recombinant. I'm just marking it like that. Okay. Now we don't know anything like that, but I'm just like I have to explain it. For that I'm just marking it like that. I'm saying these two are non-recombinants and this like a hypothetical case I am saying. So if it is so, now I said the strategy for selecting recombinants is replica plating. We have to do a replica plating work. So what do you understand by saying replica plating? You know what is replica? You are just making a copy of this plate on another plate. For example, like there is a particular device known as a replica plating device. It's simply we can say a wooden block only. It is covered with velvet cloth, and it can be sterilized also. So when we just put that replica plating device on your first plate, the colonies will be absorbed onto that particular device. Like I said, it's covered with velvet cloth. So then you will just make a copy of that on another plate. So after overnight incubation, you are observing some colonies on your first plate. Now you are transferring these colonies onto another fresh plate. These colonies are transferred here. See, obviously all the 4 colonies are transferred, but after overnight incubation, you will see only 2 colonies. Why is it so? Actually, this second plate is made with nutrient agar plus tetracycline. So only those cells which are tetracycline resistant can grow on this plate. And we know our recombinant cells, they are having only ampicillin resistant genes with them. Why? Because you inserted your gene of interest into the tetracycline resistant gene of the vector. So its reading frame got impaired. So there is no chance of producing a protein from tetracycline resistant gene. That protein only is responsible for inactivating the antibiotic tetracycline. But now that gene is got disrupted, and instead of that particular gene, protein like gene of interest is present there, and from that your protein of your interest will be produced. So now your recombinant cells, they carry your recombinant DNA molecule. They cannot grow on this particular plate because of the presence of tetracycline. But your non-recombinant cells can grow on this plate also. Why? Because they do not have any insertion on this particular site. So they have viable ampicillin resistant gene and tetracycline resistant gene. So they can grow in a medium containing both ampicillin and tetracycline. But your transformed cells, your recombinant cells, they can grow only on, they are resistant only towards ampicillin. This particular gene is intact, but this gene is got disrupted. So recombinant cells will grow on the first plate, and both recombinants and non-recombinants, they can grow on this plate, but on the second plate, only the non-recombinant cells will grow. Recombinants cannot grow on this plate. And as it was a replica of the previous plate, you can just trace the position. See, this is a colony which is non-recombinant. That's why it is formed on, the colony was developed on the second plate. This and this. These two are present on the second plate. Matlab they are tetracycline resistant. And we know that tetracycline resistant cells are missing on this. These two are missing. Matlab they are recombinant cells only. They cannot grow on this plate. Why? They do not have a viable tetracycline resistant gene. Okay. So now the task is you have to selectively pick up these two colonies, because these are the colonies of your interest. In the second plate you will see only these two colonies that they are of not your interest. You have interest on only these cells. So you will go for picking up these colonies, and this will be transferred to fresh culture media, and there selectively these cells will be grown. Now as these are well separated single colonies only, so when you pick them up and subculture them in your cell culture, now only recombinant cells are there. So now you have made a cell culture that is going to propagate your recombinant DNA molecule, and obviously your gene of interest is getting propagated. And if the proper expression signals are kept in that vector, definitely the gene of interest will code for the protein of interest, and you can go for a commercial startup of that particular product. So that is pBR322. Selection principle is insertion and inactivation of this particular gene. Insertion and inactivation of tetracycline resistant gene only helped us to select the recombinants. And now selection strategy is replica plating. Replica plating needs 2 days. First day you will make a medium with ampicillin. Second day you will make a medium with tetracycline. In the first plate you will see both non-recombinants and recombinants, and in the second plate you will see only non-recombinants. So here I will mark it as non-recombinants, and recombinants will be selectively taken up from the first plate. You can trace the position because it was a replica. Exactly the same thing was copied here, but the colonies of only non-recombinants could be developed here. These two couldn't develop here. Okay. So now we can see, like, I hope it is clear. If you have any doubts, you can ask me later. So now we can see the advantages and disadvantages of this pBR322. I said this is the simplest and the most popular. What makes it popular, that we can see? Its size is very small. Like we discussed in very big detail this particular point earlier. There are 4 advantages of keeping the size very small. I hope you remember those 4 points. Anyway, this molecule is only 4.3 kb in size. Even with the addition of 6 kb foreign material, your pBR322 will be still in manageable size. We learned that the size of a vector molecule, ideally it should be kept as less than 10 kb. So even with the additional 6 kb foreign DNA, still it is a very tiny molecule only, and we will get all the 4 advantages of keeping it small. Then the second point is selection process is easy. Why selection process is easy? देखिए, 2 selectable marker genes are there, and this will help us to find out the recombinants from non-recombinants and non-transformants. The process is very easy. Then the third point is reasonably good copy number. What is the copy number? 15 to 20 copies per cell. So this is fairly good copy number. Then approximately 21 restriction sites. Like we have 21 different sites. Here you cannot see 21 different sites. I have marked some 3 plus 4 sites. Some 8 sites only I have marked here for simplicity. So 21 sites are there. You can use any one of these 21 enzymes. 21 sites, 21 different enzymes can be used to cut open this molecule. So 21 sites are there. That is another attraction. Then the fifth point is raw material for many other vectors. This is one of the primary viable vector molecule. This is used as a starting material for the construction of many other sophisticated vectors. So now we can see the disadvantages. See, it's just like, we sometimes say optimistic and pessimistic approach. Like we say, after seeing the bottle, like half filled bottle, an optimist will say it is half full, and a pessimist will say it is half empty. Same is the case here. The same points are said in a different way. Like we are okay with this, but there are some problems. They get disadvantages. Here I said reasonably good copy number, and here I am saying copy number is not sufficient to study gene function. Like if you want to simply propagate your DNA molecule, like if you want to just test it, like whether the system will work or not, for that it's fine. But if you want to go for a commercial production, you want to purify protein from the cell culture, in that case this copy number is not sufficient actually. Like it will be very difficult for us to isolate protein. So, and if you want to study gene function transcription, translation, these two processes. So if you want to study gene function, this copy number is not sufficient. Then here I said approximately 21 restriction sites are there. Here I am saying another aspect of that. Only 11 restriction sites are present in selectable marker gene. Now I hope you understand this point. Only 11 restriction sites are present in selectable marker gene. What is the need of keeping your restriction sites on selectable marker gene? That only will help us to find out the difference between recombinants and non-recombinants, or that only will make us to find out transformants and non-transformants. So selectable marker genes, when we insert our gene of interest into the selectable marker gene, it will change the phenotypic property of the cell who uptake it. So this is very important. Basically, we will use the restriction sites which are present on selectable marker genes. You can insert your gene of interest into this site also. There, like that, many other sites are there throughout this genome. Here I have just marked one for making you understand. Here you can insert your gene of interest. But the problem is that how will you select, how will you know that like your bacterial cell has uptaken a plasmid with your gene of interest? It's difficult because there is no change in phenotypic property. Here the phenotype is determined on the basis of antibiotic resistance. If it is not in a particular like a gene which is quite important for the survival of the organism, then there is no point of inserting your gene of interest onto that. Why? Because you won't be able to select the recombinants. Or like there are options to find out the recombinant cell then too, like if we go for this colony hybridization techniques based on the gene of interest, definitely you will be able to find out the recombinant colonies, but it is again the process is very tedious. Here this selection process is very simple. With the help of antibiotic resistant genes you can find out the recombinants from non-recombinants and non-transformants. But definitely we will study this aspect later, like in library construction we have to discuss this kind of things. Like if it is not on selectable marker gene, there are ways to find out the recombinant colonies, but generally we prefer to make the process simple. Okay, now the selection scheme is lengthy. Here we said selection process is very easy, but the selection process is easy, but here it is lengthy. It doesn't mean that easy doesn't mean that it's not lengthy. Here selection scheme is lengthy. Process is very easy, but it will take time. For a molecular biologist, for a genetic engineer, time is very important. 48 hours, it's something very big. So we will there are many vector systems that they help us to find out the recombinants within a day. So this is like selection scheme is lengthy. Then segregational instability. This point is very important. Segregational instability. So what do you mean by segregational instability? In the previous slide you have seen that numerous many number of recombinant DNA molecules are there in a particular cell, and we know whenever the cell goes for division, the host chromosome, one copy of both the progeny cells will be getting one copy of your the main chromosome. In the same way, we just hope that our plasmids will also be segregated among the progeny cells in a justifiable manner. But most of the time, you know what is happening. As the number of molecules are many, it's a matter of chance. Some cells will be among right after replication, division, you will be getting two different cells. One cell is getting more number of plasmids and the other cell is getting very less number of plasmids. So this is gradually upon subsequent culturing what is happening, upon subsequent cell division what is happening, there will be a population of bacterial cell devoid of plasmid. That chance will be there. And if you do your culturing work in the absence of selection pressure ka matlab kya hai? You have to put ampicillin in the medium because only those transformed cells are having ampicillin antibiotic resistance. So if any cell is there without plasmid, they have to be killed. So for that you have to put ampicillin in the medium. So if you are not putting ampicillin antibiotic in the medium, there is a chance that both transformed cells and this non-transformed cells both can grow in the culture medium. That should not happen because most of the time if it happens so, then the non-transformed ones will overpopulate the transformed ones. Transformed ones, nothing here I mean your recombinant cells are transformed ones only. Transformed cells are of two types, recombinants and non-recombinants. But in the beginning itself we selected those recombinants. Now those recombinants are getting divided, they are growing and the cells are dividing and only their progenies are there. But if the plasmids are not equally distributed every time, what is happening? Finally there can a population can arise that is devoid of your plasmids. That we should not allow. So this segregational instability is one problem with your pBR322. Actually, in your naturally occurring plasmids, for example, fertility plasmid, it is a single copy plasmid only. For ensuring its correct distribution, once two copies are made for that particular plasmid, then it will be that the cell will be divided and one copy will be given to one cell and the other copy will go to the other cell. So like that, for ensuring this, like both the fertility plasmids, if it goes to single cell, that won't be it won't be a correct way. So for ensuring the proper segregation, there are some sequences known as PAR sequences, PAR, PAR sequence. PAR sequence. This PAR sequence will be kept in the sophisticated vectors to ensure its proper segregation into progeny cells. But these PAR sequences are not incorporated into this basic model. pBR322 is not carrying a partition sequence. PAR stands for partition. That sequence is not there. So we just depend on that chance. All the cells will be getting at least few number of your plasmids. But what's the problem? Sometimes it will be we cannot justify this distribution. Like some of the cells won't be receiving your this plasmids. So like that, a group of cells, a population of cells without plasmids can be developed there. And then host range limitations are there. This particular origin of replication that is taken from PMB1, this particular origin of replication doesn't favor wide range replication. In certain cell types only, this will get replicated. This molecule will become viable only in certain cell types only. That is another problem. Okay, so these are the advantages and disadvantages. I just want to say one thing. Like here I said it has good copy number. So most of the time what I have seen in the disadvantage, you will write here you will write it has good copy number. Here you will write it doesn't have good copy number. And again, selection process is easy. Here it is written selection process is easy. Here you will write selection process is difficult. Please don't write like that. Already I had given the explanation for all these points. That explanation must be like that, like selection process is easy, selection process is difficult. Reasonably good copy number, not good copy number. Please don't write like that. There is certain situations are there. On that basis, we are talking about these disadvantages. Okay, so this is about your pBR322. The next vector system is pBR327. pBR327 is derived from pBR322 only. And we deleted 1089 base pair fragment from pBR322. Thus we got pBR327. 1089 base pair matlab approximately 1 kb. So by deleting 1 kb DNA, we got two advantages. pBR327 has two advantages. The first thing is its copy number is increased as some 30 to 45 copies. Then it facilitates biological containment. All the segments like origin of replication, ampicillin resistant gene, tetracycline resistant gene, all these things are just like pBR322 only. But the difference is some part, like some 1089 base pair fragment is deleted. It has got deleted from this origin of replication. It's near to origin of replication segment only. Because of that, the property of this origin of replication has got changed. It has got relaxed replication control. Now it can replicate up to some 30 to 45 copies. So this is something very important. Why? Because I have already said that the copy number of your pBR322, it's not that great to study gene function. Now this is better than, the copy number is better than pBR322. The second thing is facilitate biological containment. We know the conjugative plasmids, they can facilitate their own transfer from one bacterial cell to another bacterial cell. But this is a non-conjugative plasmid. Your pBR322 is a conjugative plasmid. Because of the deletion of certain region from this pBR322, now it has got converted as non-conjugative molecule. It helps us in biological containment. So what is biological containment? It is the prevention of escape of viable cells from the experimental system to the natural environment. We do not want to see that our constructed molecule is becoming the part of the natural flora. It will have, we cannot predict the impact of such escape of viable molecules. So just by deleting this 1 kb DNA, we could change its conjugative property, and now it is a non-conjugative plasmid. So in both these ways, pBR327 is better than pBR322. And the size is also reduced, like 1 kb is lost it's become again smaller, and that is also good only because we know it's easy to work with small molecules. So the first two vector systems, pBR322 and pBR327, got over here. Next class we will study about Puck series vector.